The original premise that was put forward and actually published in The Lancet was that this was a naturally occurring virus with an animal host that got transmitted to people, but yet there's not been a single animal model found with this. Neither has there been a single coronavirus that has this PRA insert.
Are you prepared to testify under oath here today that COVID-19 is a bioweapon?
Yes, I am.
First, I'd like to cover some of your qualifications. You have an impressive 90-page resume. I'm not going to go through that, but very briefly, are you a medical doctor?
Yes, I am.
So therefore, you have an MD degree, as it's known.
I do, yes.
And what is your area of specialty?
Nuclear cardiology, and internal medicine.
And do you also belong to professional medical associations?
Yes, I do.
Are you a fellow of the American College of Physicians?
Yes.
And are you a fellow of the American Society of Internal Medicine?
Yes.
And you also hold a PhD?
Yes, I do.
And in what field?
Physics.
And you also have a law degree, otherwise known as a JD?
I do.
And are you an inventor in the medical field?
Yes.
And very briefly, can you explain the type of medical inventions that you've invented?
The ability to image the body and determine what types of disease processes are going on by looking at metabolism and blood flow.
And do you hold patents for your inventions?
Yes, I do.
Is it fair to say you're familiar with the world of medical patents?
Yes.
You have the ability to read and interpret medical patents?
Yes, I do.
And are you also an author of professional papers?
Yes, sir.
And approximately how many?
Somewhere between 400 and 500.
And do you also serve on the editorial board of professional journals?
On editorial boards and review boards for the journals.
And did you also look at the genetic code of the SARS-CoV-2 virus and especially the spike protein?
We did, as well as Professor Luc Montagnier, who is the gentleman who discovered the HIV virus, and several other individuals that we note in the book.
And based on that review of the genetic code, that informed your conclusion that this is a bioweapon?
Right. So the Biological Weapons Convention Treaty states that any adjustment or modification of a biological agent like this virus that doesn't provide a benefit for mankind is a biological weapon. These particular changes in this virus, including the PRRA insert, which are amino acids that were inserted that are very critical for the furin cleavage site for this virus to infect. The insertion sites that were made with HIV and simian or ape-like HIV equivalent viruses, the prion-like domain at the top of the spike protein, as well as the HIV glycoprotein 120 insert that Shi Zhang Li put in early on that's critical for the attachment of this virus to cells, all of which are not naturally occurring.
So, to be clear, genetic changes were made to the spike protein in a lab to create what is known as the COVID-19, correct?
That's what all the data shows.
Was that research designed to determine how to increase the spike protein binding capacity to the human ACE2 receptor?
So that was her goal of that research, was to increase the ability of the virus to infect human cells.
Now, interestingly, I think in this published research, they discovered that the naturally occurring spike protein from horseshoe bats was unable to bind to the human ACE2 receptors.
Right, that's exactly right.
So as of 2010, scientists, including Shi Zhang Li in Wuhan, China, knew that naturally occurring horseshoe bat coronavirus spike proteins could not infect humans.
That's right.
Now, this genetic code for the furin cleavage insert, the PRRA, is that found in any other coronavirus in nature?
None.
It's not found in any other coronavirus that exists in the wild.
Right. There's absolutely no other coronavirus that has this PRA insert. And that's 12 nucleotide bases, not a single base like sickle cell, but 12 specific nucleotide bases to provide for 4 amino acids that very specifically is connected with the furin cleavage site, which the U.S. government owns the patent on. Since mutations occur one nucleotide base at a time, you would have to come up with some phenomenal explanation for why 12 nucleotide bases suddenly inserted themselves into a virus that none of the other viruses in that category have. The original premise that was put forward and actually published in The Lancet was that this was a naturally occurring virus with an animal host that got transmitted to people, but yet there's not been a single animal model found with this. Neither has there been a single coronavirus that has this PRA insert.
And somebody took pieces of the genetic code of HIV and put it into SARS-CoV-2?
That's what she's angrily admitted to in publications that she had, yes. Where everybody is kind of coming up to speed with a virus and how challenging it can be to deal with. What's even more challenging to deal with are prions, because prion diseases are relatively new in our understanding for treatment, and they cause permanent damage unless they can be reversed. And so the development of a prion, putting HIV glycoprotein 120 in there, attaching to the sialic acid receptor, there's nothing about that is advantageous for people. And the ability to produce prion diseases is quite a weapon. And so the premise of gain of function really has a good premise behind it. The original idea being that if you could stay a step ahead of a potential infectious agent, say a coronavirus, if you could stay just a step ahead, you could do a good job of providing treatments for people and knowing when and where it was going to spread. This isn't a step ahead. This isn't a single nucleotide base. This is a spike protein that has an HIV glycoprotein 120 inserted into it. This is a spike protein that has 4 amino acids to make a furin cleavage site, the PRA insert. This is a spike protein that has multiple other, according to Luc Montagnier, HIV and SIV. This is not staying a step ahead of it. And making this provided no advantage for human beings, which makes it a Biological Weapons Convention treaty violation. It's plain God. And then when you say, well, where's the scientific evidence that it's naturally occurring? There's no follow-up to that. So that for people that are claiming that this is a scientific discussion, I would encourage them to stay with the fact that this is a scientific discussion. The National Institutes of Health and National Institutes of Allergies and Infectious Disease have now admitted, not that they needed to, but it's nice for them to admit that a gain-of-function research was funded for by the U.S. and that Fauci's statements that they weren't involved in gain-of-function research was not correct.
I think that it's really a fulfillment of a dream that we had together with my leadership team when we started in 19. The first week we met in January of 19 in California and to set up the goals for the next 5 years. And one of them was by 2023 we will reduce the number of people in the world by 50%. I think today this dream is becoming reality.
CBS Health Watch, sponsored by Pfizer. Anderson Cooper 360. Brought to you by Pfizer.
ABC News Nightline. Brought to you by Pfizer.
So it's really a purpose-driven company. And
making a difference. Brought to you by Pfizer. CNN Tonight. Brought to you by Pfizer. Early Start. Brought to you by Pfizer. Friday night on Aaron Burnett Out Front. Brought to you by Pfizer.
This Week with George Stephanopoulos is brought to you by Pfizer.
Today's countdown to the royal wedding is brought to you by Pfizer.
And now a CBS Sports Update brought to you by Pfizer.
Meet the Press Data Download. Brought to you by Pfizer.
On how to find the hidden sugars in the American family diet. Sponsored by Pfizer.
Bill Gates' advice on how to combat mistrust in science.
This portion of CBS This Morning sponsored by Pfizer.
You don't get vaccinated just for yourself, but also for others. You do it for all of society. That's what I said. Today, this turned out to be complete nonsense. In a COVID hearing in the European Parliament, one of the Pfizer directors just admitted to me, at the time of introduction, the vaccine had never been tested on stopping the transmission of the virus. Was the Pfizer COVID vaccine tested on stopping the transmission of the virus before it entered the market?
Regarding the question around did we know about stopping immunisation before it entered the market? No. We had to really move at the speed of science to really understand what is taking place in the market.
Ms. Small, again, we had to really move at the speed of science. Now, what I'd like to do now is pause and explain to you exactly what the speed of science means. But I haven't got a flipping clue. I do know not what it is. It's just words without meaning. The speed of science. It's just words without meaning. What does that mean? What's that supposed to mean? What's it supposed to convey? Just has no meaning to me at all, unless I'm a bit stupid, of course. If Miss Moore wants to come on the channel and explain it, then that's absolutely fine, of course. Maybe I'm just being a bit thick here and I don't understand what the speed of science is. But I've done two science degrees and a research degree, and I've never heard the term. So anyway, there's always need to learn new things. Only been doing science since I was 18.
Millions of people worldwide felt forced to get vaccinated because of the myth that you do it for others. Now this turned out to be a cheap lie.